Assalamu alaikum. This is Muhammad Hassan Shafiq and I welcome you all to the ICB online program for grade 12 biology. We are continuing with the unit 23 of chromosome and DNA, and in today's lecture we will be talking about a very important topic regarding DNA called gene expression. In gene expression we will be talking about transcription today, and we will be also talking about the genetic code. Now, by now, we all know and is also proven that DNA is the genetic material which can replicate, and this material is in the form of a specific sequence of nucleotides. The DNA inherited forms traits by which protein synthesis can be directed. So the DNA actually acts as a catalyst and catalyzes specific chemical reactions in the cell. So by now it is we know that it is a main hereditary material. It is inherited by an organism, and when inherited, it forms certain traits which actually direct the synthesis of a protein, and which will later on catalyze different reactions in a cell. Thus, a gene itself is expresses itself in a protein, or it can also express in an enzyme, since enzymes are also globular proteins that control the development of a specific character or function. Like obviously, and this expression needs to be carefully coordinated and regulated as well. We can say that proteins are linked between the genotype, which is a genetic complement of a cell, and the phenotype, which is the form or appearance of a cell or a trait. Gene expression or protein synthesis includes transcription and translation. Okay, so how gene gets expressed and protein synthesis, how it takes place, can includes actually two steps, which are actually the uh, main which is actually the central dogma in molecular biology, which means that the information transferred from DNA to direct a certain protein, or the synthesis of a certain protein in the cell, is one of the major or most pivotal points in molecular biology. The idea that DNA makes proteins via an RNA intermediate is called the central dogma, like I just said, of molecular genetics, and involves transcription and translation. In today's lecture, we will be only talking about transcription, but I will definitely cover translation as well in the next topic, in the next lecture. Transcription is defined as the synthesis of RNA from DNA, and it is the first step of gene expression. It occurs in a G0, G1, and G2 phases of cell cycle, and requires only one enzyme to be completed, like the RNA polymerase. However, it is a continuous process, unlike DNA replication, which occurs via the leading and lagging strands. There is no such lagging, and the process is quite continuous, which and involves a major single enzyme, RNA polymerase. It is a continuous process, and for convenience, we can divide it all this process into three phases: the initiation, elongation, and termination. So the process of the RNA or synthesis from DNA can be divided into initiation, elongation, and termination. Moving on to the first phase, which is initiation. Transcription begins with the binding of RNA polymerase at the promoter region, and this promoter region is a regulatory region in the gene that comprises binding sites for the attachment of RNA polymerase. So in prokaryotes, these are TATAAT, or also called TATAT minus 10 sequence. Like it's like just 10 nucleotides before your original gene sequence. We can also say upstream your gene. And TT-GACA, or TTGACA, also called the GACA box, and the TATAT sequence is also called the TATA box. In prokaryotes, this TATA box is actually at the -10 sequence, and the GAGA box is actually -35, which is 35 nucleotides before your original gene sequence. This sequence in eukaryotes is at -25 for the Tata, and CAAT, the cart box, at the -71. Name of these sequences refer to, like I just said, their position in the promoter region, that these sequences are located approximately how many nucleotides before the transcription start point of the gene. Like obviously, we will not want our polymerase to sit on the gene, because it will not transcribe those regions on which it is sitting. So definitely it has to be somewhere upstream of your gene, which is a sequence in the DNA called a promoter sequence, and it has a specific nucleotide sequence which will be recognized by the RNA polymerase, where it will bind and then initiate the synthesis of an RNA intermediate from DNA. Now, the major transcription enzyme, the RNA polymerase, consists of 4 subunits. But just before that, let's go to figure 23.10, where we can see the start point, and which is labeled +1. Just before the start point, we have a -10 sequence and a -35 sequence, and all these sequences, the start point, the Tata and the GAGA boxes, are actually in the promoter region of your DNA. So transcription will start at the +1 side then. Like DNA polymerase, RNA polymerase can also initiate RNA synthesis from the 5' to the 3' direction, which we will come in a while. Now, let's talk about the structure of RNA polymerase. RNA polymerase consists of 4 subunits, 2 alpha and 2 beta subunits called the core enzyme, while the sigma factor is required for RNA polymerases to bind on the promoter. The core enzyme and sigma factor together constitute a holoenzyme. So the core enzyme consists of 2 alpha and 2 beta subunits, but the sigma factor, when added to this core enzyme, gives you a complete holoenzyme. Once RNA polymerase is attached on the promoter, the sigma factor is removed and the core enzyme catalyzes the remaining process. It is similar to the DNA polymerase in that it also adds the nucleotide to the 3' end of a growing strand, like I mentioned earlier. But unlike DNA polymerase, it does not require a primer to perform its polymerase activity. So there is no primer needed here. In prokaryotes, only one type of RNA polymerase is found, while in eukaryotes, we have polymerase of 3 times, namely RNA polymerase 1, which synthesizes ribosomal RNA, RNA polymerase 2, which synthesizes messenger RNA, and RNA polymerase 3, which synthesizes transfer RNA. As the RNA polymerase binds to the promoter, DNA duplex becomes unwinded, base pairs are broken down, and a bubble-like structure, the transcription bubble, appears. So as the RNA polymerase binds, the sequence of changes which happens is that the duplex unwinds, obviously, because RNA polymerase needs to bind to a single strand. So in that case, the bonds between the base pair also has to be broken, which forms a bubble at that initiation site called the transcription bubble. This ends the initiation here, where RNA polymerase is ensured and properly binds to that region. The second region is called the elongation phase. And in this phase, as the RNA polymerase binds to the promoter, it begins to arrange and polymerize the ribonucleoside triphosphates or ribonucleotides complementary to the template strand of the DNA. So it starts adding RNA nucleotides complementary to the DNA sequence, and it does not require a primer, like I said, to initiate uh, polymerization. One of the 2 strands of the gene act as template for transcription. Like I said, that any of the 2 single strands can be used as templates. Uh, the template strand is also called the antisense strand because the messenger RNA which is formed during this transcription process is complementary to this strand. So the template is also called the antisense strand because the RNA formed will obviously be complementary to the template. So the sense will also be opposite, which is why the template strand is, we are calling it the antisense strand. The other strand of the gene is called the coding strand or sense strand because the sequence of the messenger RNA in this case will be exactly similar to the DNA strand of the coding strand, where the only obvious difference will be thymine being replaced by uracil. So in that case, we have one antisense strand and one coding strand. In the elongation phase, the RNA polymerase keeps on moving from the 5' to the 3' direction of the RNA towards the terminator region of the gene. Besides, its transcription bubble also keeps moving along the DNA, leaving the growing RNA strand protruding out from the bubble, which I will show in 23.12 on page 195 on the next slide. The event continues till the RNA polymerase reaches the termination region of the gene, and this is what I was talking about. This giant pink structure is the RNA polymerase, which keeps on walking on the DNA, and as it keeps walking, the RNA strand keeps protruding out of the transcription bubble. The third phase is the termination phase, which is the last phase of transcription, where the gene stops the synthesis of messenger RNA. The terminator region consists of a series of GC base pairs followed by a series of AT base pairs. The part of messenger RNA which is transcribed in this region projects to form a loop-like structure called the GC hairpin, followed by a small tail of poly-U nucleotides. This GC hairpin causes the RNA polymerase to stop the synthesis of RNA. So it actually forms a big resistance to the movement of RNA polymerase on the DNA, forming a pin which will actually stop RNA polymerase from further traveling on the DNA, stopping the process. Now we will, like you can see on figure 23.13, how this GC rich hairpin loop is formed, followed by AU sequences. And once this is formed by complementary base pairing within the single stranded RNA, it will stop RNA polymerase from further moving, in, terminating the process. And the final RNA will then be released from the will be released into the cytoplasm. But before being released into the cytoplasm, it has to be modified in order to ensure its safe journey to the ribosome for protein synthesis. And for that, it needs to undergo certain modifications, which we will talk about in our next topic. The post-transcriptional modifications of messenger RNA in prokaryotes, like there is no delay between transcription and translation. As the messenger RNA emerges from DNA, it undergoes translation due to the absence of a specific nucleus. But in the presence of eukaryotes, there is a definite presence of a nuclear envelope through the pores of which the messenger RNA has to travel. So, but it has to, so this pre-messenger RNA formed has to be modified into a mature or functional messenger RNA, because transcription occurs in the nucleus while translation in the cytoplasm, and messenger RNA has to move from the nucleus to the cytoplasm, during which it has to keep itself safe from enzymes like phosphatases, which might digest the phosphate group, and nucleases, which can further degrade this molecule. So eukaryotic messenger RNA also contains some non-coding region called introns, which need to be removed and spliced during the process. Post-transcriptional modifications involves two steps. Number one, addition of a cap and a tail to protect it from degradation, and RNA splicing to remove non-coding sequences. So number one is that the addition of a cap and a tail to the molecule, and also the second modification involves the removal of unwanted structures called introns, which need to be spliced or cut out. A cap in the form of 7-methyl GTP is linked to the 5' end, and a tail called the poly A tail having a chain of about 330 to 500 adenine nucleotides are attached to the 3' end. These two modifications prevent the DNA to be degraded by phosphatases and nucleases. The removal of introns and maturation of primary messenger RNA to secondary or functional messenger RNA is called RNA splicing. Splicing is catalyzed by the spliceosomes, which is a large RNA protein complex. So the major complex which is responsible for the removal of unwanted sequences in the pre-messenger RNA are called the spliceosome. Later on, the spliced exon fragments are joined together with the help of the RNA ligase enzyme. Now, moving on to figure 23.4, you can see the unwanted introns, which are then removed, the addition of a cap and a tail cap of 7-methyl GTP at the 5' end, a poly A tail at the 3' end, and the spliceosome, which is in a pink spherical balls, cutting out the introns in this diagram, and the introns exercise can then be seen. This later on modifies the pre-messenger RNA into a mature messenger RNA, which is now ready to leave the nucleus and enter the cytoplasm for protein synthesis. Genetic code is the relationship between amino acids sequence and nucleotide sequence. We do know that a gene is a sequence of DNA nucleotides which carries information for a synthesis of a certain protein. Therefore, in order for amino acids to be made, the relationship between the amino acid sequence and the nucleotide sequence is very important. And the code in the DNA which actually carries this sequence is the genetic code. In other words, we can say that the information in DNA for the synthesis of protein is called genetic code, which is transcribed or copied into messenger RNA. The messenger RNA consists of a random sequence of only 4 kinds of nitrogenous bases: A, G, C, and in the case of RNA, U. Now the question is that how can a code with 4-letter alphabets specify a proteins at any given point containing only one of 20 different amino acids? Clearly, a single base cannot specify an amino acid. If one base pair specifies a single amino acid, then we will be carrying only 4 amino acids in that case. And the proteins containing only 4 different amino acids will be formed. Now, nor it is feasible for 2 because they will end up in 16. But if or 3 base is sufficient, with 3 base pairs of 4 into 4 into 4, we get 64 combinations. Each is specific for a particular amino acid. Now, obviously, if we are having 64 codes and only they are specifying 20 amino acids, we will later on see that one amino acid may be coded by more than one codon. So what is a codon? This is a sequence of 3 bases along the length of messenger RNA that specifies the particular amino acid. Let's look at this in figure 23.15. We can see the 20 amino acids and they are coded by more than one codon. Some are definitely just have one, but some have as much as 6. Like we can see arginine having 6. We can also see leucine having 6. So, and the ones which are having only one are methionine. We can also see tryptophan. The stop codons are the ones which stop transcription, and if they are present on the messenger RNA, the transcription will stop, which are UAA, UAG, and UGA. Now, there are a total of 64 codons, out of which three are the stop codons, which are also called nonsense codons, which they do not code for any amino acid. And AUG methionine is the start codon, which directs the start of the uh, um, which codes for the start of the amino acid chains. Now let's talk about the properties of the genetic code. Genetic code has the following characteristics. Some amino acids are coded by only one codon, while some may be coded like leucine and serine up to six. This property is called as code degeneracy. Okay. Code degeneracy means that the code is not as strong as it appears to be, since we are seeing different codes carrying specifying the same amino acid. Now, one of the most important point which I have pointed out on my slide as well is that the genetic code is universal. It is the same in almost all organisms. For example, AGA specifies arginine in bacteria, humans, and all other organisms whose genetic code has been studied. Because of the universality of codons, the genes can be transferred from one organism to another successfully and translated in their new host as well. The study of genetic code of mitochondrial DNA, however, shows that genetic code is not quite universal, because UGA is normally a stop codon in mammals and humans, but in mitochondria it is read as tryptophan. AUA was read as methionine instead of isoleucine, and AGA and AGG for termination of protein synthesis instead of arginine. So it is not quite universal, as mitochondrial DNA does appear to have some exceptions. There is no punctuation. The reading frame is continuous. It means that there is no gap between the codons. And the fourth one is that the genetic code is non-overlapping. The messenger RNA sequence AUGAGCGCA is not read as AUGUGAGAG. It will be read as AUGAGCGCA. So it is non-overlapping, right? So this was all for today's lecture. We will continue with the translation and other topics in the next one.